We're here with Dr. Ken Stuart of the Center for Infectious Disease Research. And Ken, can you just tell us a little bit about CIDR?
Yes, I'd be happy to. So the center is a private not-for-profit research organization that is entirely focused on global infectious diseases. The biggest killers, frankly, and based on health problems. They are malaria, HIV, TB, and a variety of other, perhaps lesser-known parasitic diseases, but also more recently diseases like Zika or dengue virus.
Interesting. So that's your current focus of your research. I'm curious about when you started CIDR back in 1976, and it was Seattle Biomed. How did you go about getting this from an idea into reality?
The center started because I was at a university in Florida. I had just gotten my first grant. I wasn't particularly happy with the university that I was at, mainly because I didn't feel there was enough ambition to do the research that needed to be done. At that time, I worked on an organism that causes African sleeping sickness called trypanosomes. It and related organisms cause diseases that kill approaching 1 million people a year. And these are neglected diseases. I recognized at the time nobody in this country was actually working on trypanosomes. It was a fascinating problem, an important problem because it was killing tens of thousands, at that time probably 50,000 people a year, just that one disease. And then the two related diseases were killing even more diseases. So I was fascinated by two things. One is the incredible biology that these organisms represented, but also the real practical human problem that these were diseases that were killing many, many people, and in fact making many more people suffer from these diseases, and then causing a lot of other problems in terms of the economic consequences of these diseases. So the suffering was not only physical suffering, but economic suffering, destabilization of the community and the cultures, and conflict, and so a number of things. And those two things really captured me. Understanding the biology of these organisms, which continually fascinates me, but also realizing that this is a practical, very important problem, one that's affecting many, many people.
So at that time, at least, you said they were neglected. Why do you think they were neglected as opposed to some of the other diseases at the time?
Very simple answer to that question. These diseases are diseases of the poor. There's no economic incentive, direct economic incentive of these diseases that would induce a pharmaceutical company to develop a vaccine or a drug, because the people who are suffering from these diseases do not have the financial wherewithal to pay for the drugs or vaccines that are needed. That's true today as it was then.
That's a sad truth, but such as it is. You had realized that you had this ambition to attack those diseases which didn't have the focus. How did you raise funds? I mean, you were from an academic background, but how did you go about making it into an organization?
So I got my first research grant from NIH when I was at this university in Florida. And they told me I was going to get this grant. But I called up NIH, literally called up NIH, the person who was responsible for administering my grant, and told them that I really didn't want to start the grant up at this university, but I wanted to start up a research center and move the grant to that research center. And because I thought, rightfully so, that I could be more successful in performing on that grant, and also that it was a better environment for me to do the research that needed to be done. I was also exploring various areas to cite such a research activity. And one of the places that I explored was Seattle area for a variety of reasons. One of which was because the University of Washington has a long and rich history in infectious disease research. It's a big research, well-funded research university. And so I thought that that would be an excellent environment, academic environment, and research environment in which to do this work. In addition, the Pacific Northwest was also an attractive environment to live in. So I made some phone calls and flew out and met somebody who had actually had similar thoughts, who was at what became the Fred Hutchinson Cancer Research Center. That person also had a grant, and so together we started the Research Institute. And we did this by basically having a colleague who became one of the initial board members go down to Olympia and file the forms for a 501c3, a nonprofit research center.
Nice.
And then I transferred the grant to this organization, packed up my U-Haul, and drove from Florida to Seattle.
Nice. That took a lot of initiative.
I was highly motivated. I think I'm still highly motivated, actually.
Well, that comes through. In the early years, though, what were some of the more, I guess, business, organizational, and finance challenges you dealt with? Because talking to other academics, one grant will not see you through.
Right. They were many. I can tell you that. When I first moved out, one of the pieces of equipment that I needed for my research was an ultracentrifuge. It cost $20,000 in 1976, so probably more like $75,000 now, something like that. I had half the amount of money to buy that. So I went to what is now defunct Washington Mutual Bank, and I showed them the notice of grant award from NIH, and I said I need to borrow $10,000. And for whatever reason, they agreed. They loaned me $10,000, and I went and bought that piece of equipment and sat down and wrote a supplement to the grant, which fortunately got funded. So it was a little bit of a risk, and or I would have been on the hook for $10,000. And then not only that, but then I was able to negotiate with the Beckman, which is the manufacturer of that equipment, to provide that piece of equipment to me at a discounted rate, and also throw in some other parts of that equipment, fairly expensive, some of which were experimental at the time. So I was able to get that going. So that's one example. We really didn't have much infrastructure, so we had to work with.
So you were literally like nailing
we built the benches. We literally built the benches that we did the research on, and we built some of the equipment. So there was electrical equipment, power supplies for electrophoresis. So at night I would buy these kits from Edmund Scientific and sit up and solder the pieces together for these power supplies, and cut up plastic and glue it together to make the containers for doing what's called gel electrophoresis. Just did everything ourselves. Even though our equipment was homemade and we were not in the most elegant of conditions, some of the findings that we made at that time, so one of the early discoveries we made is in an area called, or a field called RNA editing. So we were one of the early discoverers of this whole process of RNA editing. We found that this totally novel process, which was how genetic information, which is contained in DNA and then made into RNA and then ultimately into proteins, which are the workhorses that do things. We discovered that this RNA, after it's made, the meaning of that RNA can get changed. And this was extremely novel at the time. In fact, it wasn't well accepted for a couple of years, I can say that.
Well, that just proves that it was novel, doesn't that?
Absolutely. In fact, I got invited to a meeting at a famous place called Cold Spring Harbor, which was where the primary RNA work was being done at the time, as this how RNA gets treated after it's made from DNA. And I went to this meeting. I was invited probably 4 or 5 days before the meeting convened. Very daunting situation, because these meetings have all the best researchers in the world go to this meeting. And when I went there,
So you were a young researcher, and they
I was
just
a young researcher, yeah.
called
we
you
just-
up and said,
Well,
we want
they
you to
had
speak at the premiere event? Like,
Yes.
wow.
Well, they had seen the publication that I had submitted, and that probably the person who reviewed that publication and was pretty shocked by the findings. When I got there just before the meeting started, which was at a Sunday evening, and I knew nobody there, zero. These were people I only knew from their publications. The next morning I sat sweating through the session while all of these other people were presenting this fantastic work. So I got up and I noticed during the presentations that nobody was asking many really interesting questions. They were all just detailed. I got up and I was introduced, and then I said, well, I'll be surprised if there's no questions after this presentation. And there were 30 minutes of questions.
a 5-minute presentation and then 30 minutes.
30 minutes of questions. So that was a very pivotal point. And I would say that most of the people who were at that meeting didn't actually believe the results that I had. They thought that I didn't know how to sequence RNA at the time, but we did. Now it turns out that this phenomenon of RNA editing, not exactly the same type, is occurring widely. It occurs in our brain. It controls the composition of neurotransmitters, neuroreceptors, and it controls our high versus low density lipoproteins. It functions in protection from HIV. There's some mechanisms that are inherent in cells that are this RNA editing process.
So you had these great findings. People didn't accept them. You didn't have the buy-in, and your grant was not going to last forever. How did, institutionally, what was the next sort of crux point?
Good things about science that's sometimes difficult for non-scientists to understand is when people don't believe your work. It's not necessarily a bad thing or a criticism. It drives you to prove it. And so we did. In fact, I tell people in my lab regularly, my job is to be a skeptic. You have to demonstrate to me that the finding that you've made or the conclusion that you've drawn is actually correct. So we published many, many more papers that amplified or extended and therefore confirmed that finding, and then found out many more things about this process of RNA editing. And we're still, 40 years later, we are still working on this process and how it's controlled and how it's integrated within the much more complex cellular environment. These cells have 10,000 genes. And so this is only a small part of this much more complex machine, this parasitic cell. So that is a normal part and welcome part of the scientific process, is to present your data, especially your early findings, which you initially are skeptical about yourself because you don't understand them, because they're new. And then others who are hearing them, they also realize, well, I'm not sure I believe this, show me. And then eventually, by doing multiple experiments, you not only find out which of your findings were correct, but then you tend to find more and more, and they build upon each other. So after these initial findings in my own laboratory was expanding and growing and getting more and more recognition, other scientists found our organization and joined us because they were also interested in having highly focused research activities in a more independent environment. So about that time we decided that the laboratory at that time was in Issaquah, which is about 10 miles and not exactly in the center of the research universe at that time.
Not exactly in the center of Seattle either.
Not in the center of Seattle. So we decided that it was time to move into the city. And so one of our board members, in fact, president of our board, was also at a sister organization called PATH, which is now just down the block from us, actually, which is really in the business of delivering health interventions to developing countries. And they were developing a building on the Ship Canal down in Seattle, and they asked us if we'd be interested in becoming a tenant in that building. So we did, and we initially moved in the basement, literally in the basement of that building.
Right on the canal.
Right on the canal, right? And then more and more people became aware of us, and more people started joining us. And eventually, we kept growing, and we grew into 3 buildings, parts of 3 buildings right on the ship canal. But there was a very pivotal point not too long after we moved to the ship canal. I decided that it was time for us to get some external evaluation of our whole research program and us as an institution. Mm-hmm. So I asked 5 very able people to come and review our programs, that get a scientific advisory committee together. Mm-hmm. So one of these people was the head of a foundation, the Borough's Wellcome Fund. Another person was at a biotech company. Mm-hmm. Another person was the dean of the School of Public Health at the University of Washington, and there were a couple of others. So these were experts. Mm-hmm. And so we asked them to come in for a day and a half, and we would present our research programs and the directions that we were going. The first recommendation was actually by the dean of the School of Public Health, was an invitation for the center to affiliate with the University of Washington. Mm-hmm. And the reason for this was, by that time, our research capabilities had not only expanded but grown in quality, that we were stronger than the basic science department, which was the pathobiology department at the University of Washington. And so the dean recognized this. Mm-hmm. And so I always like to say that because they invited us to affiliate with the university, that actually it's the university that's affiliated with us.
Rather than the other way around.
Rather than the other way around, right. Mm-hmm. So I ended up chairing that pathobiology department for 7 years. I was instrumental in developing a PhD program there, which is called the pathobiology program. It's a PhD program. It's that pathobiology department has now become fused into the global health department, which spans the School of Public Health and the School of Medicine. And we still at this institution get graduate students from that department, as well as from other departments, the Department of Immunology, for example, or, and other departments.
So it sounds like you were really able to grow Seattle Biomed into, you basically did a ton of outreach to local university, the science community, yes, and just kept promoting both your research and your ideas about what should be researched. And then...
Yes, so it wasn't all my ideas. Yeah. So we would, as a faculty, so we had grown at that time to about a total of 100 people, something like this. And so we would periodically get together, have a retreat or meetings, and decide, okay, what are the next directions that we're going? What is our strategic plan? We had a board. We've always had a board, and our board who are people with very good business expertise, said quite early on, well, you need to have a strategic plan. That was a learning exercise in itself. How to actually effectively develop a strategic plan is something that I had no experience with, but have done multiple times now. And so as part of this strategic planning, we developed, I guess what I would lightly call an algorithm of what diseases would we work on and why. So the diseases that we chose were diseases that were big, important diseases. Diseases where we had the expertise that we could actually do something to advance understanding of these diseases that could lead to vaccines, drugs, and diagnostics, that there were resources out there that we could identify and get to support the activity, and that we had researchers that we either had them or we could recruit researchers to work on these diseases. Mm-hmm. And so in over several years, not that many years, of course, even though we started on these neglected diseases, trypanosomes, leishmania, and those related diseases, mm-hmm, the next disease that we chose was malaria, mm-hmm, which is, as you know, a huge...
Because you wanted something easy and simple to attack, right?
Quite the opposite. I am perversely attracted to biological complexity. Okay, it's one of the things that I find intriguing, is trying to solve these complex problems. So I went over to NIH and to the laboratory of malaria, the malaria laboratory at NIH, which was run by Lou Miller, a very famous malaria researcher who's still at NIH. And I asked him to give me a culture of malaria because I wanted the institute to start working on malaria, because I was frustrated listening to the science and I knew this was important. So I brought, literally on the airplane, a culture of malaria. So I
Like a little petri dish, or like
little plastic bottle,
plastic
little
bottle.
culture bottle with live malaria, human malaria. Couldn't do it today.
This was before you would get searched.
Yes, yes, right, yes. So I brought it back to Seattle so I can say I brought malaria to Seattle. And I don't know if I'm the first.
Distinction.
Yeah, I don't know if I'm first, but I brought it to Seattle. And I had a postdoc who was working on something on trypanosomes, and there was a related problem in malaria. And I said, I'd like you to work on malaria. And that was the beginning of our malaria research program.